Okay, we're back for another video about, this time, chapter 2.3, carbohydrates and lipids. So we're going to start with carbohydrates. We've already kind of looked a little bit at these. Carbohydrates are molecules that always have this relationship between the three compounds that exist that make them up, or three elements that make them up: carbon, hydrogen, and oxygen. So there's always the same amount of or number of carbon and oxygen atoms, but there's always twice the number of hydrogen atoms. So and this N is basically how many of these carbon atoms you have, and they can vary between 3 and 7. I think in most cases it's 5 or 6. They can form these ring-shaped molecules, and the monosaccharides are the smallest version or the simplest versions of carbohydrates that exist. And here we have some examples: glucose, galactose, fructose, deoxyribose, and ribose. These are the ones you tend to find. Well, you find all of them in food. However, these are the ones we use as energy, basically, and these ones we use more for DNA and RNA. So they are used as building blocks more, and these ones are used more for energy production. So the simplest form are these monosaccharides, and in general, we can put monosaccharides together into more complex types of carbohydrates, and that's a general kind of phenomenon that you can take simple units and build more complex units. And we tend to then in general call the simplest building blocks you can use to build more complex things as monomers. So monosaccharides are the monomers of carbohydrates. We're going to get back to this more later. So we can see that we can put two monomers to form a dimer of carbohydrates called disaccharides. So disaccharides are the specific form of dimer for carbohydrates. And here we have 3 examples. So sucrose is the type of disaccharide that you find in table sugar. And we find it in, for example, in sugarcane and sugar beet. It's supposed to be beet, of course. Sorry. Lactose is also a type of sugar. That's the milk sugar found in milk from mammals, like cows and humans. And maltose is a type of sugar found in germinating seeds from what? It's supposed to be wheat. Oh my god, I made so many spelling mistakes. From wheat and barley. So in beer, for example, you find malt sugar. Sometimes you might be able to, or you might be requested to look and understand the structure from this kind of drawing. So here we see the unfolded version, for example, of fructose. So you see the carbon, and oxygen in the red, and then you have all the hydrogen molecules, atoms here. And the middle one looks then at 2 glucose molecules put together. So you see that they're bound together here. So now we have an actual disaccharide, and this would be a monosaccharide. And this is another type of disaccharide than the lactose. So it's I'd say this should be our glucose and this should be our galactose. So that's another way of looking at it. And you can put, of course, more than 2 together to form polymers of carbohydrates, or polysaccharides specifically. And these can be put together in many different ways. So here we have, for example, 3 or 4 examples of polysaccharides. And if you look at here, we have the different shapes of them. You see that they can be very different. If you hear some weird noises in the back, it's my cat running around. She's going crazy. Anyways, so each of these little circles or balls are small monosaccharides. So the difference between these ones is basically how you put the molecules together. Like here you see, if you put them in opposing fashion like this, then they can form this very straight kind of polysaccharide structure, and that's what we find in cellulose, and it makes it very densely packed and robust, and that's why it's so good in the cell wall of plants. Amylose here is a type of starch, so you have the sugars put together in a different way, and that means that it's gonna form this repeating helix structure. And in another type of starch, you put the sugars together in kind of perpendicular patterns sometimes, and it starts to form these branching structures. And in those are the starches of, for example, in the seeds and so on of plants. So this is the kind of polysaccharides that we can eat. This one we can eat it, but we're not gonna be able to digest this one. And in animals, we also store monosaccharides as polysaccharides in our liver and in our muscles. So those are also branched, just like this type of starch, and form these very interesting patterns. Glycogen then is the name of the one in animals. So these are 4 types of polysaccharides found. The 3 first one in plants, this one in animals, but they have very different properties depending on how the molecules are put together to form either very rigid structures or something that's maybe more easy to break down. So these are mostly used for or accessible for digestion. So this is used as a building block instead. And they can also look like, you can also visualize them like this. This is our glucose molecule, and you see in cellulose it's put together like this, and alternating, flipped back and forth, and then you form these straight patterns. This is this type of starch, the amylose type of starch, that forms then these very curved structure, and in glycogen we see these branched structures instead. And when you put these together to form the bonds between them, you actually, every time you do that, for example, here we have two disaccharide and 1 monosaccharide. And you know that there's on this monosaccharide here, there's a hydroxyl group facing this end on, and this one you have a hydrogen atom here and a hydroxyl group over here. So when you fuse these together, you're basically removing these ones, this hydroxyl group and this hydrogen, to form the bond between this monosaccharide and this one. So these 2 are removed, and you see that there's 2 hydrogen and 1 oxygen. So it means that every time we put these together, we form water. And when water is formed on a glass, for example, we say that water condenses, becomes liquid again. So this, when we generate polymers from simpler structure, we say that the process called condensation, because water is always formed. And the opposite then, when we break polymers down into the constituent parts, we have to use water to kind of reform these hydroxyl groups and hydrogen atoms. So we always need to use water to break down polymers into smaller parts. So this is called hydrolysis. Lysis is to break stuff apart, and hydro for water. So breaking stuff down with water, basically. I hope that makes sense. So condensation, putting them together; hydrolysis, breaking them apart. So this happens both in carbohydrates, and it's also how it happens, for example, I think in lipids as well, but definitely amino acids when you put them together. Okay, let's look at lipids then. So lipids are a little bit more tricky. Lipids are not as easily kind of grouped together and defined as carbohydrates, but this is the main definition, that is compounds with an oily or greasy consistency that are also insoluble in water. And in general, they contain carbon, hydrogen, and oxygen atoms, just like carbohydrates do. But sometimes they have other things as well. And there are very many different types of lipids. So phospholipids that we looked at before, they are lipids. But you also have other types of lipids like cholesterol and let's see, ergosterol. Oh my god. These are what we call sterols. You also have fatty acids, and in animals and living organisms we store fats as triglycerides. And they have, as you see, quite different kind of structure to them compared to, and are more diverse than carbohydrates. So, but we can look at, for example, triglycerides first. We see that they are formed by an alcohol, glycerol, and 3 fatty acid chains here. And we see that they are also formed by condensation. So yay, I was actually correct. But fatty acids then that make up the kind of main part of perhaps of the triglyceride are these long chains of carbon atoms with hydrogens bound to them. And at the end we have this carboxyl group that we talked about in this lesson before the break. So it's a carbon with a double bond to an oxygen and another oxygen with a single bond, and to that one you have a hydrogen atom. So this is the acid part and this is the chain. So let's look a little bit more at fatty acids. So they can vary in their length, so be long or shorter or longer. But it can also vary in the number of double bonds they had. So this is a saturated acid, so it has no double bonds. And you see that it's very straight. However, you have also, this one is an unsaturated fatty acid, and then it has a double bond. And this is a polyunsaturated fatty acid, so it has many different bonds. And the last thing that they can vary is if on the 2 sides of the double bond, if the hydrogens are on the same side, it's a cis bond. We call it. Cis means that they're on the same side, or trans if the hydrogens are on different side of each other on the different sides of the hydrogen bond. So cis fatty acids, then what happens to the structure? Well, it means that actually the whole thing becomes a little bit bent, because now you have all this extra space here where the thing will move into. So if you look at the 3-dimensional structure here, you see that cis fatty acids are bent, whereas trans are straight. So both saturated fatty acids and unsaturated fatty acids are both straight, and that means that you can pack them more densely together. So lipids that have mostly saturated fats become more dense. So the type of, like butter for example, those are mostly have saturated fatty acids, and it means that it has a more solid consistency in room temperature, whereas things that have cis unsaturated fatty acids, they are mostly then oily and liquid. It can't be packed together, so they become less dense and become oily in room temperature. And we can see then that the fatty acids, for example, on triglycerides can be either unsaturated or saturated. And we see the same thing here we've talked about before. And what we have been able to, or a lot of science, or a lot of lots of experiments and observations suggest that it's healthier for people to eat more food that has unsaturated fats in them, and specifically cis unsaturated fat. Trans fats are rare in nature, but they form if we, for example, try to harden oils, for example in cookies and so on. We form these trans type of fatty acids when we try to harden fats. But I think we're much better at removing them now. This was a bigger deal, I think, back in the days, that you would have cookies with trans fats, and that would be bad for you, because our blood vessels in normal function, they have their look like this, nice and pinkish. However, when the fat contents in the blood is a lot of saturated fatty acids, it appears that that we're more likely to build up fat in the arteries. And specifically, it's not the saturated fatty acids themselves, it's actually cholesterol that's built up. So one would think that eating cholesterol means that your cholesterol levels in the blood would also increase, but apparently it's not so simple. It appears instead that if you eat lots of saturated fatty acids, you get the types of cholesterol, you increase the levels of cholesterols that are more likely to form this kind of thickening of the arterial wall with fat. And when that happens, you get higher blood pressure, and sometimes they can also rupture the kind of wall of this artery, and you can form a blood clot or thrombus. And that can then happen in your heart, in the vessels on top of your heart, and that can give you a heart attack because blood can't pass through here anymore. And then these tissues here, which rely on the blood supply here, would not get anything and they would die. That's when you form a heart attack. But there are lots of studies that also challenge this view, but so far most of the recommendations from food and health organizations is that you should eat more more oily things instead of more fatty things. So basically that would entail the cis fatty acids are what you find in nuts, in seeds, and in olive oil and so on, and less saturated fats that you find in animal products. So try to get more oils into you. What do we use the lipids for? Otherwise, phospholipids, of course, are important for our cell membranes, but animals also use triglycerides to form, for example, blubber in mammals in the ocean to insulate our bodies so we don't lose heat as easily. It's also important on land, but it's very important for animals that dive very deep into the ocean where it's cold. Sharks use fats as they have quite low density in their livers to regulate their buoyancy. And one of the main parts of what we use fats for is to get energy. So this is slightly complex, we're going to get to this later, but basically the body can use carbohydrates, fats, and proteins to generate ATP, the energy, the currency molecule of cells. And the amount of energy that you can find in, for example, carbohydrates and proteins, this is the energy unit kilojoules per gram, is virtually the same. However, in fat, you get more than twice the amount of energy per gram. So it's a much more effective or efficient way to store energy. So that's why fat is so important. And that's all we had to say about carbohydrates and lipids. We'll see if there's going to be a video about proteins or if we're going to see each other in person. So I hope you enjoy these videos, and I'll see you later.